Good day and welcome to your virtual science class. I am your teacher, Miss Jihan Simon. This is Chapter 7, Lesson 4 from your Pearson Realize book. Today we are going to see traits and variations. Diversity of life. Organisms from the same species tend to have many similarities. The northern elephant seals show that very different traits can exist in two individuals. This is figure 1. Seals of the same species share most of the same DNA, yet there are differences in their appearance. This male on the left and female on the right relax near the ocean. Others are invisible, such as type 1 diabetes or sickle cell anemia. Differences have the potential to be passed on from one generation to the next and change the population. Any difference between individuals of the same species is a variation. Two friends with different eye color have a variation, green or brown, of the same trait, the eye colors. Variations may be due to DNA inherited from the parents, exposure to certain environmental factors, or a combination of both inheritance and environmental factors. Variations can be helpful, harmful, or neutral. Examples of helpful variations are like the owl butterfly on the right. They express this. Consider a population of butterflies avoiding predators. Some have the same wing color pattern as a poisonous species, or some of them can look like the owl butterfly. When this variation is passed from one generation to the next, the offspring are more likely to survive and reproduce. Examples of harmful variation are the ones who threatens a population's survival. For example, low blood oxygen levels can be found in individuals with sickle cell anemia. But scientists have recently discovered that the sickle cell anemia makes some people immune to malaria. So we are still not sure if this is a harmful variation or if the body adapted to make the red blood cells immune to malaria in some places. And examples of neutral variations, such as different eye color that do not benefit or harm the population. You received 23 chromosomes from your mother and 23 chromosomes from your father. The combination of genes found in these chromosomes codes for the proteins that determine your traits. As you can remember when we saw fertilization in the last lesson. Types of chromosomes. There are 2 types of chromosomes found in every one of your cells. Of the 23 pairs of chromosomes, 1 pair is sex chromosomes, while the other 22 pairs are autosomal chromosomes. Sex chromosomes are the pair of chromosomes carrying genes that determine whether a person is biologically male or female. A human female inherits 1 X chromosome from her mother and 1 X chromosome from her father. A male receives 1 X chromosome from his mother because his mother is going to always pass an X chromosome to the offspring. And the father is going to decide the sex. So the male is going to receive a Y chromosome from the father. This is the activity that we have made from your book, Punnett Square, where we determined the probabilities of this couple, the father on the top, where it says X and Y, and a mother on our left, where we'll inherit X and X. So the probability of this couple in their offsprings is that they will have 2 female and 2 male probably. Types of mutations. Genetic mutations. Many mutations are the result of small changes in the organism's DNA. Just one small change to a base pair is a mutation and may cause an incorrect protein to be made during protein synthesis. As you can see in figure 4, there's 3 types of genetic mutations. In the first DNA sequence, we can see a normal DNA sequence with all its bases and in the correct order. In figure 2, we are going to see deletion, where one base is removed, one of the base pairs is removed. In this case, the last TA has been deleted. In the next figure, we're going to see addition, and if you can notice from the original DNA sequence, we now have an extra base pair. In the next figure, we have substitution, where one base pair is switched from another one. And we have sex-linked mutations, which a mutation can occur on any chromosome. Some mutations occur on sex-linked genes, which are genes carried on a sex chromosome. Because the X chromosome has more genes than the Y chromosome, most sex-linked mutations occur on the X chromosome. Many sex-linked mutations are recessive. Hemophilia is a recessive sex-linked mutation where the individual's ability to clot blood is reduced. Males are more likely to exhibit hemophilia because they have only one X chromosome. So we can see here how hemophilia is inherited to the offspring. In this case, we have on our right a pair of parents, a father and a mother. The father is going to inherit X and Y. The mother, which is a carrier of hemophilia, is going to inherit X1 and X2. When fertilization occurs, we have these probabilities in which one son is going to inherit X2 from the mother, which is a carrier, and Y from the father, which is a healthy human. And on the next figure, we have a daughter who inherited X1 from the mother and the healthy X from the father. So she is not going to express the hemophilia because the X that the father inherited is going to make the job that the X1, which is a deficient or abnormal gene, cannot do. I mean the protein synthesis. And on the next figure, we are going to have a son which inherited the X1 chromosome, which is the abnormal, and the Y chromosome from the father. This son will express hemophilia because he has the abnormal chromosome from the mother. And since he was inherited a Y chromosome, there is no other X chromosome that can synthesize the proteins adequately for him. So he will have hemophilia. And the next one is a daughter, which received two normal chromosomes, two healthy chromosomes. On the left, we have another pair of parents. This time, the father is the carrier, the carrier of the gene. He is expressing hemophilia. So the offsprings, he is going to have a son which is going to inherit X1 from the mother and Y chromosome from the father. But this son is going to be healthy. He is not going to carry or express the abnormal disease, the abnormal genes. Then we are going to have a daughter who's going to be a carrier because she inherited the X chromosome from her father. Next, we are going to have another healthy son. And next, we are going to have another daughter who is a carrier. So, as you can see, it changes a lot depending in which of the parents is the carrier of the gene. If the father is the carrier of the gene, we are going to have two daughters who are going to carry the gene. And if these daughters have sons, they might be able to express hemophilia. Environmental factors. Interactions with our surroundings and the conditions in which we live have the potential to change the way genes are normally expressed. First, environment factors can change nucleotides, the building blocks of nucleic acids, DNA and RNA. Secondly, the chemicals found in DNA can be changed. Organisms come in contact with harmful chemicals and radiation on a regular basis. These agents are called mutagens because they can damage DNA in such way that it causes mutations. Some mutagens naturally occur while others are synthetic. For example, radiation in the form of ultraviolet or UV or X-rays are naturally occurring mutagens. We can see from figure 6, damage from sun exposure. UV radiation from the sun harms skin cells. UVA radiation penetrates into the deep layers of the skin. UVB radiation penetrates only the top layer of the skin. Please complete this activity and all the rest of the handwritten activities in your book and upload your images into a Word document and submit your work into Google Platform for Friday. Synthetic mutagens can be found in pesticides, asbestos, and food additives. Gene expression. Changes in the way genes are expressed may occur naturally or because of the environment. An example of natural change is when a caterpillar transitions to a butterfly. As the organism develops, the DNA does not change, but the genes are read and expressed differently. This means the same animal is going to have the same genes, the same DNA, but this time they are going to have different functions. It's not the same functions as a caterpillar than as a butterfly. The environment can change the way genes are expressed. Identical twins have the same DNA, but can acquire different traits when they grow up in different environments. Activities such as smoking and unhealthy eating habits can also alter the way genes are expressed, which changes a person's traits. Mutation effects. Mutations can be helpful, as you can see this fish which is camouflaged into the coral reef. They can be harmful mutations as lung cancer or cystic fibrosis, or neutral mutations like color of the eyes or hair. Mutations in reproduction. Not all mutations are the result of small changes in organisms' DNA. Some mutations occur when chromosomes do not separate correctly during the formation of sex cells. A sex cell can end up with too many or too few chromosomes. When this happens, either additional proteins are created or too few proteins are created. This is called non-disjunction, where the cells stay together as the cells divide. We can remember the normal meiosis and mitosis processes in which our sex cells go through. And we usually end up with meiosis in two cells with two different chromatids. But when non-disjunction occurs, sometimes we are going to end up with two cells that have more than one chromatid or less than one. Karyotypes. Sometimes doctors suspect that an individual has a genetic disorder based on observable traits. To accurately determine if an individual has a chromosomal mutation, a scientist will create a karyotype. You can see the picture on your right. That is a karyotype which a genetist can perform. And this karyotype is from a normal human being. We are going to be comparing more karyotypes as we pass on slides. A karyotype is a picture of all the chromosomes in a person's cell, and then arrange the chromosomes by size and matching chromosome patterns. Homologous chromosomes are paired to provide a quick overview. As you can see, these are many different types of karyotypes. The first karyotype is from a healthy female. The second karyotype is from a female which has an abnormal disease called trisomy 21 or Down syndrome. This occurs because during non-disjunction, the cell will end with more than two chromosomes. Number 3 is an example of a Turner syndrome or a monosomy X. And number 4 is an example of trisomy 18 or Edwards syndrome. Please complete the activities in your book. As you can see here, X monosomy, Edwards syndrome, Down syndrome, they all have different observable traits that can make doctors suspect about a genetic disease. Protein changes. Amino acids are the building blocks of proteins. Proteins are considered the architects of cell function. A change in the amino acid sequence can alter the directions for protein synthesis. Jumping genes may cause a species to change. This is figure 10, and it is the example of a squid who is discovered to have a changing RNA. And scientists suspect that this jumping genes or changing in the RNA of the squid can help the squid population to change. Please log into our Google Classroom and complete your assignments. Thank you and have a great day.